Okay, so good afternoon, dear colleagues. I'd like to welcome you to this analytical lecture, and thereafter there is another event. We will have the presentation of the IUFA Lifetime Achievement Award together also with a lecture. So I'd like now to introduce the reason why we have this analytical pharmacology lecture. It's a distinguished award of IUFA that was founded by Sir James Black following the 12th International Congress of Pharmacology in Montreal in 1994. And this quadrennial recipient is elected by the IUFA Nomenclature and Standard Committee, which is led by Steve Alexander, and it recognizes investigators who have made significant advances in identifying, defining, and exploiting novel drug targets. So just to give you a figure, who were the earlier 6 recipients, it were Alberto Kaumann, David Kulkon, Terry Kennekin, Paul Van Hoot, Arthur Christopoulos, and Brian L. Ross. And the distinguished award of the analytical pharmacology lecture is now going to Professor David Gloriam. Thank you very much for joining us here, and I'd like to introduce him. David is a professor in Computational Receptor Biology at the University of Copenhagen, where he leads the research cluster for GPCR function and drug discovery and a Pharmaceutical Data Science unit. His group runs a GPCRdb database where 4,000 researchers each month to retrieve reference data and access online tools for analysis, visualization, and experimental design. He has got his PhD from Uppsala University in Sweden, including the bioinformatic identification of 24 novel human G protein-coupled receptors. He later identified physiological hormones of such undercharacterised orphan receptors and functional probes for a range of receptors. He made 2 postdocs in the UK at the EMBL-European Bioinformatics Institute in GlaxoSmithKline. In 2018, he joined the University of Copenhagen, where he has received the ERC Starting Grant, Lundbeck Foundation Fellowship, and Novo Nordisk Foundation Ascending Investigator Awards. So David is a corresponding member of the Nomenclature Committee of the International Union of Pharmacology, IUFA, and he's one of the coordinators of recommendations to describe ligand bias towards signaling probes and safer drugs, and his group recently developed an online research of biased ligands and pathways effects to advance the bias signaling field. And now it's your, David, I'm happy to listen to you.
Thank you very much, Ingolf, for the kind introduction. Thank you to the organizers for such a fantastic congress again. And thank you very much, of course, to IUFA's Nomenclature Committee for awarding this keynote and award. Yes, as Ingolf already mentioned, today you will hear about computational GPCR pharmacology in this analytical pharmacology lecture. I've attended 2 last previous World Congresses, and I've had a pleasure to listen to, in my eyes, what were role models when I set out and started my career. Both Brian Roth and Arthur Christopoulos, great pharmacologists, among the recipients of the analytical pharmacology lecture. So I'm much honored to stand here today. I hope to tell you something about computational pharmacology and something about GPCRs, even if you don't know anything about this beforehand. So I've tried to adapt and to share why it's so exciting to work on these, especially in these times. And just one slide of background to introduce what is a GPCR. It's a G protein coupled receptor that couples to G proteins. It's a kind of signaling microprocessor at the interface from the extracellular environment, sending signals into the cells. It does so upon mediation, the effect of about 1/3 of approved drugs, as well as 2/3 of hormones or neurotransmitters in our bodies. And there's about 800 of these receptors, so you can imagine it's quite abundant signaling network. And of course also intracellularly, I mentioned the G proteins, but beyond that, other transducers that directly bind and engage with the receptors, such as arrestins, GRKs, and others. In computational pharmacology then, what is that? I think it can be many things, but at least what I'll show you and tell you about today is computational drug design, some of the data science or data analysis where we try to increase our understanding of receptor structure and function, as well as infrastructure, online databases, and actually tools on the internet where you can go and find research, maybe actually do some of your research. And when I go to conferences, often someone walks up to me I haven't met before and say, I love GPCRdb. And that's another great honor, of course. GPCRdb, I think, is the major hub for G protein coupled receptors, has about 50,000 users in the field. And now we've extended this with additional databases for G proteins, arrestins, and biosignaling. And I'll tell you a little bit more about that. It's not just databases, it's actually research tools. Of course, you go there to find reference data, but you also go there to analyze data or to visualize data. In some cases, also, data-driven design of new experiments. So with the clicks of a few mouse button clicks, you can actually design binding site mutations based on all the structural interactions, as well as more than 34,000 mutations from literature. And you can deposit data to increase the dissemination, such as these type of mutagenesis experiments, as well as biased ligands. And now to the outline of this talk. So today I will try to share some of the major data revolutions that's been in the past. So I'll make a look back, but also look forward toward outlooks. So I'll give an example of computational GPCR pharmacology studies for genomes, structures, and G protein couplings, as well as online databases and tools, quickly, as well as an outlook. So if we've seen these breakthroughs recently, if we look forward in the coming 10 years, what are the current major challenges for the field? So let's start out with genomes. And of course, in the early 2000s, when I was about to start my research career, the human genome had been sequenced. And President Bill Clinton said this will revolutionize the diagnosis, prevention, and treatment of most, if not all, human diseases. Fantastic. And soon thereafter, of course, followed many, many more genomes and initiatives, not just genome assemblies, but also connecting phenotypes clinical data and phenotypes. We also see that non-human genomes have vastly increased, and this is just taken from the NCBI Genome Data Viewer and their eukaryotic genome assemblies. And this gives many new research opportunities. You've heard about AlphaFold. It uses co-evolution of residues to find interacting residues in the protein folds, to mention one. But as I mentioned, I started my research in the early 2000s, and I felt so lucky that I was born into these times and that I could start a research career, because clearly to me, at the time, this was the most exciting time to do research. Because we could go on a treasure hunt, we could take the known receptors, and all of a sudden, very quickly, we could identify a great number of new receptors from the human genome sequence. And in the course of my PhD in the group of Helge Bischot, we managed to identify 24 new receptors. And they were quickly picked up both by academia and industry. There was a large interest. But it left us with another research problem. Now that we have all these receptors, which are called orphan receptors. So if you think of a child that is orphan, it doesn't have a parent. If you think about a receptor that is orphan, it doesn't have a known physiological ligand, and typically not a known function. So we have all these receptors. We know that other members of these receptor families are very important for our physiology as well as drug targets. So there's a huge research problem in de-orphanizing these receptors, meaning identifying the endogenous ligands that activate them, presenting possibly new drug targets or even peptides that would constitute starting points for drugs. So, which physiological ligands to look for? In one of our studies, we looked back at the known GPCR ligands, and we found that the majority of these are peptide or proteins. In another study, we also looked at the trends in GPCR drug discovery. We found that there was an increasing focus on peptide and protein drugs. So we decided we want to try to identify peptide ligands in this study. But how to do that? Learning from our experience of identifying the receptors from the human genome, we realized that, of course, the peptide ligands, they should also be found in the genome. They come from precursor proteins. There are genes in the genome encoded just the same way as the receptors. And additionally, they have some unique properties. They are cleaved, and they have to be secreted to the outside of the cell to bind to the receptor. So we hypothesized that peptide ligands can be identified from the genes and the cleavage and the secretion. And where to go and look for these? We started out with actually the human proteome, so the canonical protein sequences in UniProt, about 20,000 at the time, as potential precursors for peptides. Then we subsetted these to look at the ones that are secreted, meaning they have a signal peptide in the N-terminus, which is a prerequisite to be secreted. And just for validation, we checked, can we find the known GPCR ligands there? Yes, we can. And then that left us with about 1,400 potential precursors after subsetting again to keep only the proteins that doesn't already have a known function. So in this case, we arrived at around these 1,400 potential precursors. They have a signal peptide, they have an unknown function. But we cannot assay the precursor. We need to identify the peptide subsequence in the precursor. And what you're looking at here is POMC, which is one of those precursors. At the top here is actually the amino acid sequence. It's too small, so you can't see it. There's this signal peptide in the N-terminus, and then in green are the different peptide hormones. And if we look a little bit closer at this, and we look at these valleys and mountains, which represent the conservation across 300 animal genomes. It's pretty easy to see a pattern. We can see that where the green peptide hormones are located, it's where there is high evolutionary conservation, so these valleys. Another thing, maybe you wonder what the gray bars are at the top. So these are precursor cleavage sites, and those have consensus sequence motifs that are recognized by the enzymes, often dibasic motifs. So if you put these two together, the conservation and the cleavage sites, we find that we can recollect potential peptide ligands based on the conservation and the cleavage sites. Well, but we also need to predict which of these many receptors are potentially receptors for peptides. And based on this, we looked again at the known peptide receptors, specifically the ones for which we had a structure. And we could see that they typically have a beta sheet in the second extracellular loop that gives a larger surface to interact with large ligands such as peptides or proteins. We also see that in sequence, they cluster a little bit apart. And in this way, we cannot distinctly say, but we can filter out a large number of non-peptide receptors. So our theory is that these characteristics can help us enrich peptide receptors. So going towards the experiment, of course, we need to prioritize the predictions. So we used machine learning, random forest, based on the characteristics I showed on the previous slides. And then we had a commercial synthesis by GenScript. And with the receptors, there were assays being set up in-house in the Hans-Braun and Osborn group. Again, these receptors were selected based on characteristics. And to make sure we don't miss any of the responses, we actually used triple assay systems in parallel, measuring different types of receptor activation aspects, and whereof 2 were time resolved to make sure we don't measure at the wrong time point. Yes. And then, yeah, computational prediction took some time, I think about a year, but about 3 years went into the hard pharmacology work in validating all these peptides against all these receptors. So this was an all-to-all comparison. And this involved, of course, the local group of Hans Brian Osborne, as well as Brian Roth and Raymond Penn in the US. So if this was a cooking program, now is where we say ping, and I would take the cake out of the oven and show the results. So we managed to find peptide ligands for 5 orphan receptors, and those are the ones listed here. And what you're looking at now is, in the first case, the peptide, and then EC50 values, color-coded, in different type of assays. And if you look closely, you will actually see that it was a good decision to use multiple assays, because some of these receptors only show an activation in arrestin-based assays and others in non-arrestin assays. So, yeah, the 2 to the right here are arrestin, and here the other case around. So all of these were validated in at least 2 assays. And then I'll show you also something where we developed an online database and an outlook for genomic data. So not peptide hormones, but genetic variants and isoforms of receptors. We developed online resources in the G protein coupled receptor database that were published as part of studies together with Madame Babou. He's an excellent data scientist, having worked a lot on GPCRs. So this means it gave an opportunity for readers of these papers to go online and repeat the analysis, or maybe do an analysis of their own based on the data from the publications. And to give an outlook, one slide, the group is currently working on trying to pair receptors again, but instead of assaying all peptides against all receptors, to do one-to-one pairing of receptors and peptides on the computer. I'm not going to tell you exactly how we're going to do that, but we will certainly use a lot of genomic data in this process. And also in terms of pharmacogenomics, the genetic variants and isoforms, Alexander Hauser, who was in my group and did the discovery of the peptide hormones, he's actually started his own group now, focusing on pharmacogenomics, and will also build an online database. So if you have an interest in pharmacogenomics on GPCRs, contact him directly. And that takes us to the second chapter of this lecture, which is to say something about the structural revolution and the impact on GPCR research. First, this is a general picture taken from the Protein Data Bank, PDB, showing the increase in crystal and cryo-EM structures per year. And you can see in the past 20 years or so, there's been a great increase. And again, here you see it as an overall on all structures. As a GPCR researcher, this was clearly the most exciting time in my life to start a research career. With all these structures coming out, I felt again, I'm so lucky, I'm so privileged to be a researcher in these times. So this is just to show from the eyes of a GPCR researcher what was going on in the early stage of this structural revolution. Every time a new structure came, we would send an email to each other. And here it says, the GPCR structures just keep coming, so keep it up. And then, of course, a description of what the structure tells us, because every structure gave valuable novel new insights and could give a breakthrough for the given receptor we're working on. And this is a complementary figure showing the increase in GPCR structures color-coded by family and taken from GPCRdb. So clearly there was a really large increase of structures. And this gave a kind of luxury problem because with this many structures, actually more than 1,000 structures today, it became a bottleneck in understanding all these things that the structure can tell us. So basically converting the structural data to an understanding, mechanistic understanding. And for this reason, we spent actually 3 years to develop a pretty advanced structure analysis platform in GPCRdb that will compare different structures to identify determinants, mechanistic determinants of structure and function. We also developed a tool to cluster structures based on their conformation, as well as a tool to design new mutagenesis experiments based on structural data. Experiments, mutations that would then stabilize a receptor either in the active state or the inactive state. So if we start with the structure comparison tool, of course you can look at one structure and what you get is the structural features observed in that structure. It gets a little bit more interesting when you have a set of structures, because then you can look at the distribution of features. And my favorite, if you have two sets of structures with complementary properties, you will then be able to identify unique and common features, maybe determinants of receptor function. And the type of structure features, it really ranges from the macro level, where you think about the folding of the protein. GPCR has 7 transmembrane helices. How do these move upon activation, for example? Down to local variation in the helices, or properties of the backbone, and the residue side chain contacts, and residue properties of individual residues. And then, of course, I mentioned these online research tools also support visualization and analysis. So these are different type of visualizations that can take a day or multiple days to do on the computer, but you do it on the online resource in a matter of minutes. The second tool is the structure similarity trees. And I think many people look at this and see it as a phylogenetic tree based on sequences, but it's not. So this tree is based on the structural similarity of receptors. And when I say structural similarity, I mean the 3D conformation of the backbone. And to avoid bias based on superposition, which changes a lot depending on the region, we've done an all-to-all residue distance comparison. And this is also how we classify active and inactive state of receptors in GPCRdb. And the last tool, which is the mutation design tool, it of course sets out with structural data that is unique to active or inactive structures, presents the different residues and the mutations to choose. But it also shows available knowledge from literature. So we've annotated more than 34,000 mutations from literature, binding site residues. We also have thermostabilizing residues and expression increasing residues. So this gives you an idea to actually validate some of these predictions before you go and do your own experiment. But now to the more scientific study where we exploited these tools. So at the time, what was known was largely activation mechanisms of class A GPCRs. That's the largest class of GPCRs. There were several studies that had looked into that. However, there are several classes of GPCRs, and at the time we had just we had just gotten access to both inactive state and active state structures for classes B1, C, and F, and major human GPCR families. So this, of course, raises the question: does the overall GPCR superfamily have the same activation mechanisms as we've already seen for class A GPCRs? Or do these other classes differ? And here we use the structure comparison tool, and we compare specifically active and inactive structures for each of these classes. And bear in mind, the features we were looking at are the ones that are unique to one of these sets, so to the active or the inactive structures. And these we refer to as state determinants. And we went all the way from the macro switches to the residual micro switches, and I'll show a small selection, give you a small overview of the major findings from this study. So first we look at the macro switches, or even the transmembrane helices and the packing. And what you're looking at here are two different receptors, inactive in blue and active in red. So we're comparing these states, and then we're also looking across the membrane. So this means that here's the extracellular surface, here's the membrane mid, and here's the cytosolic end. And that's what you see here. These are the 7 helices numbered and their location. And the arrows will show you the movement from the inactive to the active state. And this is something we did then for all receptors that had a structure both in the active and inactive state, and we collected a meta-analysis. And just to give you a very high-level overview, we see that these movements correlate very well with what one can expect from the function of receptors, which is we see the largest movement on the intracellular side, because that's where the very large effector proteins bind. We see an intermediate movement on the extracellular end where the ligands bind, and we see a much more stable and more rigid receptor at the membrane mid. Then we also want to see if there is a state determinant or a helix macro switch that is universal for all the GPCR classes. And when we compare this meta-analysis again, because here you actually see all 4 classes in the same circle, we see that one helix stands out, and that's TM6, especially on the cytosolic side, it moves out. And this is, of course, common knowledge from class A GPCRs, but we can now see it moves in all classes to give room for the transducer proteins. But what is then the mechanism of this switch? And here we actually see a much larger diversity. What has been commonly known, and I think emphasized in the field, has been the tilting of TM6. And we see that in classes A, B1, and some in F, not so much in C. We see a slight lateral movement instead in class C. However, there's also a little bit underappreciated rotation of TM6 that also can increase the distance between many residues. As well as in class B1, the secretine family, there is an unwinding of TM6 that is unique. And I also want to make a remark that in class C, it goes to the end here in sub-bullets B and C. In class C, they form dimers. So 2 receptors will come together, and the major rearrangement upon activation is actually the rearrangement of the dimer rather than the individual bundle. So all in all, TM6 tilt, that was a known mechanism, is not a universal mechanism, but TM6 moves in different ways. And then we continue to the microswitch levels and to the different contact networks between residues. And that's what we see in 3D here for the 4 different classes that we studied. We see the overall number of contacts. We also see how many of these contacts that are inactivating or activating, because they stabilize preferentially one of these states. And what we see is that the classes differ a lot in how many of these restraints they have, stabilizing the states. And maybe also, as could be expected, a majority of contacts in the classes are inactivating, which explains, of course, very well why GPCR is normally inactive until activated by an agonist ligand. And I know there's a lot of data in this figure, but it's also a very interesting figure. So I'll tell you a little bit about what this tells us. Here we try to map these state determinants that we identified from the structures onto mutagenesis data and structural interactions with ligands and G proteins. Where you see text in a circle, that's an amino acid that is a state determinant. Where you see gray or orange, that is a residue that interacts with a ligand or a G protein, and the strength of color means more of those interactions across receptors and their structures. And where you see a border around the residue, that denotes a more than 5-fold effect from mutagenesis experiments. And what we can conclude when we look at all this data is that ligands and G proteins do interact directly sometimes with state determinants. So stabilizing receptors in an inactive or active state by a direct interaction. However, most determinants are found in a transduction pathway that goes in between the ligand binding site and the G protein binding site. To validate these predictions or observations, we engaged in a collaboration where we did alanine mutations and tested the activation of GS and G15 via the beta-2 receptor stimulated by adrenaline. And what we could see is there was a change in potency. There was a large difference between the predicted determinants versus non-determinants, but not in Emax. And when we look at where these are located, we see that nearly all of them cluster in the transduction pathway between the ligand and G protein. We also see that they tend to form intrahelical contacts. So they are part of the stabilization of different helices. And now a short outlook also for the structural field. You can't talk about structures and outlook without mentioning AlphaFold, so I'll start with that. AlphaFold, of course, gives access now to kind of all proteins and structures, a highly accurate prediction. So also for GPCRs, we can now cover all GPCRs by simply downloading a structure model. We can also cover both states, so there's a version of AlphaFold that actually uses the state definition from GPCRdb to pre-select the templates and thereby get different models of the same receptors in the inactive and active state. Also, there's another version of AlphaFold that can model complexes, such as especially genetically encoded proteins, such as the peptide ligands or protein ligands in complex with GPCRs or the transducer proteins, G proteins and arrestins. And the reason I'm mentioning all of this is now available also in our online resources or coming very soon. So you'll be able to have a look at these structure models and use them maybe to design your own hypothesis for experiments. Another advance we're seeing, of course, goes into virtual screening, which is the process where you simulate what goes on in our bodies. So how a drug molecule binds to a receptor cavity, forms interactions, they're shown here with dashed yellow lines. And why do this on the computer? Well, the reason is you can now buy billions of compounds and you cannot test all those compounds even if you have a high throughput screen facility. We can screen all of those compounds on the computer, select maybe 100 compounds that will then go and assay and identify ligands. And it looks something like this. We'll throw all these different molecules into the binding site, and then of course we'll have to assess which are the best interactions and to score these ligands. And I've been very excited about two studies from where virtual screens have been performed by the labs of Brian Choquette as well as Seva Katrish, where one of the major findings is that when they go and test much larger libraries, they call this ultra-large libraries, they can yield very potent and selective ligands already out of the computer. So shortcut drug discovery by reducing the resources and time needed for drug optimization. So this is very, very exciting. And one of the things I'd like to do going forward and receive some grants for, but I don't have any data to show, is trying actually to predict, based on only the 2D structure of a small molecule, what will be the mechanism, what will be the selectivity for the receptor, the pathway, as well as the modality of a ligand. And the way we want to do this is, of course, similar to what I showed in the scientific study. We can store data on the residue level in our database. We can combine data from pharmacological experiments and structural interactions in our database to make these models, which will, of course, use both the data-driven approaches we normally use as well as machine learning. But also the prospects of identifying peptide ligands, or not so much identifying, but building them residue by residue ab initio based on patterns in the receptor residue binding sites. So I believe this is something many groups are working on, but I also believe in the GPCR arena we have a unique data infrastructure and an ability to exploit advances in data science and machine learning. And that takes me to the third chapter today, which is about the data we've recently received in GPCR G-protein couplings and how we have designed a common coupling map to integrate the different data sets. So I've only mentioned, I think, my databases so far, but I'd say a much larger database and a true inspiration is the IUPHAR BPS Guide to Pharmacology database. We have a collaboration and we actually present some of the data from this database also in GPCRdb. And this is the largest resource for G-protein couplings still, and for a very long time was the only resource for G-protein couplings, annotated from literature. However, recently, the development of biosensors enabled large data sets of G-protein couplings to be published. First, a TGF-alpha shading assay by Aska Ino, and then a so-called GEMTA assay by Bouvier Lab. And there are also many additional assays that can do the same things. They just didn't publish those data sets, but I'd like to mention, for example, the groups of Brian Roth, Nevin Lambert, and Kirill Martymyanov. Anyway, now we have all these free data sets, and that presents us with a problem. Which data to use? How does the data compare? And if I want to know the coupling, which data set should I go to? Can we integrate this data somehow? And this is what we tried to answer in this study. And we started by looking at just simply the distribution of data. And if we look at the coverage simply of the GPCRs and the three different data sets here, referred to as Bouvier, Ino, and GTP for Guide to Pharmacology, and we look a little bit closer at this Venn diagram, we'll see actually the two very large new data sets contributed only 11 new GPCRs that didn't already have coupling data in GTP. However, if we look at the G-protein coverage, we see that for the same GPCRs, there's many more G-protein couplings. So couplings that were maybe missed in literature and not yet characterized. And especially, there's a very large increase in the G12-13 family, and I believe this is because robust assays have not been available that long for this G-protein family. So we conclude that the new studies added relatively few GPCRs, but many new G-protein couplings. But how many of these couplings are novel then? And we define a novel coupling as not being in the Guide Pharmacology database, but being supported by both of the two new large-scale studies. And we quickly concluded that leaves us with 38 GPCRs and 101 couplings on the individual G-protein level. So there is still quite a bit of novelty in these data sets. How do we best unify these coupling data sets? So that's what I mentioned as one of the research problems initially. We defined supported couplings in this study as being supported by two data sets at least, or all three. But we made an exception for Guide Pharmacology because normally when a coupling is annotated in the Guide Pharmacology database, it's supported by multiple articles already. And based on this, we devised a common coupling map that used log Emax over EC50 as a common measure of the Emax and EC50 values that had been reported in the publications. And also qualitative couplings on the G-protein family level where there's only the Guide Pharmacology data. And we avail this data and this common map in the database, GPCRdb, and I don't have a slide on that, but you can browse this data and put any filter you want in terms of the support, and you can also get access to the underlying data from each of the underlying data sources. And now that we have this common coupling map and we have this data, we want to learn how selective or promiscuous are GPCRs and G proteins. And G protein selectivity is one of the big questions still in the GPCR field. And when we just did a overall analysis, which is what I selected here, and we look at the number of G proteins for GPCRs, or the G protein families, because there are 4 G protein families, we see that the vast majority of GPCRs are selected for 1 or 2 of these G protein families, rather than promiscuous for all of them. And if we turn this around and we look at the number of GPCRs for G protein, so you have the different G protein families on the y-axis and the number of GPCRs here, we see that there's a large difference where some G protein families are very promiscuous for receptors. So GIO family has the largest number of coupling receptors, whereas G1213 seems to have the smallest number. And actually this box should have been placed at the bottom, so we're now at the final of this presentation, where we're going to look at the online resources and also an outlook for couplings. And here I'd like to mention ligand bias, and there's been actually a session about bias signaling on GPCRs, but just in case you didn't have a chance to go there, I'll try to give a quick definition. So ligand bias, that is when different ligands bind to the same receptor but engage different transducer pathways. So this is a mystery. How can they do that? Because the ligand is on the outside of the cell, the G protein is on the inside of the cell, they never meet. But still, via this same one receptor, they can do a unique pairing. It does so by conformational selection. It means that the same receptor has multiple active states. So in the previous chapter, I told you about inactive versus active state, but actually there's multiple active states that have their own conformation, and this conformation then selects for different agonist ligands and different transducers. So we made an online resource, we call it the Biosignaling Atlas, where we present guidelines as well as things you can use to do your experiments, such as reference ligands, as well as a very accessible Excel file template to calculate ligand bias. Of course, bias ligands annotated from literature, pathways and the physiological effect or a potential therapeutic benefit of a given pathway, as well as the opportunity to deposit data. So if you publish a biosignaling study, you can also disseminate the data in the Biosignaling Atlas, and thereby more people will find it, and you can compare it to all the existing wealth of data. Also, I want to have one slide just quickly on guidelines for ligand bias, because as you all know, pharmacology is very complex, and the more you work on it and the more research is done on it, the complexity just continues to grow. And therefore, we've seen, especially in the ligand bias field of GPCRs, there's been problems sometimes in reproducing studies that others have done, or get a clarity of actually what was done when you read the article. So for this reason, we publish guidelines for the design and reporting of ligand bias. And who are we? Well, it was coordinated by, I think, a much larger name in the field, Terry Knacking, who is one of the previous lecturers of the analytical pharmacology lecture, as well as me and Peter Kolb, as well as a large number of pharmacology experts. And it was published, of course, via IUFAR. And I'm not going to go through the guidelines. I'm just going to give you a direction. Exceptionally, unusually, when you open this paper, if you're busy, go directly to the supplementary information. Because here you will find that the first 2 appendices are checklists that list all the recommendations, both for the design as well as the reporting of ligand bias. And a very short outlook for this field. I think this is clearly the most exciting time of life to do research, because now ahead of us, there's a real grand challenge with a potential huge impact both for pharmacology and drug design. So we need to identify selective pathway probes for all these receptors. They need to be selected both for the receptor and the pathway, and they need to have the right modality. And if you think, and then you need to map the pathway effects. So what does this pathway mean for physiological function signaling or for therapeutic prospects and targeting? And when you think about scale of this, all these at least 300 plus non-sensory GPCRs and all the different transducers that directly engage with the receptor, this is a daunting task. But then, of course, the potential of designing drugs that can have a therapeutic benefit by isolating the therapeutic pathway over potentially adverse pathways. And this I presented before, virtual screening, but I want to add that in last year, 2 additional virtual screening studies were published where candidate drug compounds with benefit in mice models in terms of side effects could be identified for virtual screening. So we're seeing advances now where computational approaches can also identify bias ligands towards safer drugs. So this is very, very exciting. And with that, I'm just actually on time for the question section, and I'd like to acknowledge all the people behind this, both the computational as well as the pharmacology groups that contributed to the results I presented today, as well as to the foundations that supported this research. And thank you for your attention.
Thanks a lot, David, for this outstanding lecture. Thanks. So it's now open for discussion. We have your microphones, and please identify yourself.
Hi, David. Beautiful talk.
Hi, Ken.
Hi. So one of the major problems in drug discovery and development is off-target effects. What do you foresee in the future for predicting off-target GPCR effects of drugs that we may have under consideration for translation?
Yep. So we have a study we will submit soon on serotonin receptors. So there's actually 12 receptors all activated by serotonin, and many residues in the binding site are conserved, but not all of them. And we're trying to map, so basically we can dock a ligand into the binding site, either in 3D by molecular docking or by other approaches we've developed to get the binding mode. And based on that, we can look at selectivity hotspots, which are then maybe not a single residue that is unique for your receptor, but maybe a combination of residues that together will be having unique interactions. Or there will be off-targets which have larger residues and thereby give a steric clash to the ligand. So this is the type of residue level information that we can store in a database, and once we have the binding mode of a compound, we can predict the selectivity of that compound.
Please.
Roberto Denucci from Brazil, great talk.
Yep, thank you.
Naive question from someone outside the field. What would you expect to happen in your models with receptor dimerization or heterodimerization?
That's a good point. So you identified a hole in my presentation. So I didn't include that in the AlphaFold structure models. We haven't tried that. We should go and try that. Yeah, I believe it will be more challenging. So I think AlphaFold is dependent both on structural templates, but also evolutionary information about co-evolution of residues. So to what degree have residues co-evolved at interface of those dimers? That would be what limits the performance of AlphaFold.
Yeah, Roland Seifert from Hannover Medical School in Germany. How do you deal with the issue of data reproducibility? Because there is variation in EC50 values and Emax values, and even qualitatively, some studies, in particular regarding beta-arrestins, cannot be reproduced, which may be due to various factors. So I didn't see any aspect in your presentation regarding this very complex issue.
Yes, thank you. Very important question. Yeah, so we have different resources in our databases where EC50 values will be reported, or IC50 values for that matter. So we have more than 200,000 ligands coming from Chembl. We have ligands from the gut pharmacology database, both endogenous as well as tool compounds. We have those mutations or binding site residues that I mentioned, etc. So where we have access to that information, we will store that information. And it will be there when you download the underlying data spreadsheet. But for simplicity in the online interface, it will not be shown, because what we often show is a meta-analysis across data sets.
Now in the back, here, please.
I think you just answered Karen's question. Hi, David, that was absolutely beautiful. I know you didn't really want to go into it, but you alluded to the fact that you're trying to do a one-to-one pairing now, where you're actually predicting a specific peptide for a receptor.
Yep.
Within the bounds of what you're comfortable telling us, could you give us a bit of an idea of how you think that might work?
Could we take that one-to-one after the presentation?
More than happy to.
I'd love to sit down with a piece of cake and or coffee with you. Yeah.
Sounds great.
Okay, thank you.
Happy with that?
Yeah.
Hi,
Michael, please.
I'm absolutely delighted you've got this award, and it's really well deserved. But I've got a couple of questions. A couple of years ago with Babu, you gave a paper which I thought was really absolutely important, which was mutations in the opioid receptor, among others, which showed that with certain mutations, you could then change completely the pharmacology of agonists and antagonists. And based on the opioid crisis at present, that strikes me as being one of the most structurally important questions of our time in pharmacology. Can you tell me how it's advanced? I've got a second question after that.
Well, I'd like to redirect that question maybe to Alexander Hauser, who was a brilliant PhD student in my group at the time and traveled to Madame Babu in Cambridge at the time to complete that study. And now he has his own group focusing on pharmacogenomics. But I couldn't agree more that it's a really important problem with the opioid crisis, of course, and the prospects of identifying biased ligands also, maybe avoiding the lethal side effects. But yeah, you should talk to Alex. I'm sure he's willing to talk to you. Yeah.
Okay. And secondly, does fentanyl, there are indications that fentanyl may have a different pose in the opioid receptor. Do you have any indications for that? Because it's a key question again, because fentanyl is obviously a ligand of major difficulty.
Yeah. Yeah, it sounds very interesting. No, we haven't tried that, but I guess there are multiple structures, so it should be feasible to do that. Yeah.
Because you had just mentioned, here, you have just mentioned pharmacogenomics. I wonder, we know in pharmacogenomics much is known in ADME genes about regulation and consequences for drug efficacy and side effects. Much less is known in receptors. We know there is a plethora of mutations, but evolutionarily, it seems so that they are not so serious that they have really a strong pharmacological effect. Is that true, or what is your opinion on that?
Yeah, this will of course be a very large generalization, but there's a large variation where some mutations will be silent and not have an effect, and other genetic variants will have a dramatic effect. Some of those effects will manifest as a disease already when you're born, others a disease you develop, others you don't notice until you take a drug, and you don't get the effect you expected from the drug, because the variant now changes the drug effect. And it's not just a smaller effect, it could be the other way around. You get a way too high effect, making it adverse.
But at least there are no recommendations so far to consider such variation in
no, and this is exactly where the field needs to be going in the future in personalizing medications. And I believe there's been other sessions at this Congress that maybe have discussed that, but there's a lot of work, and it's already starting to rule out. And then maybe another way into the future, maybe even preventive medicine. So you profile before you get a disease, and you know where you need to put in the effort in a healthier living.
Okay. Thank you. There are further questions, please?
Yeah, thanks for a great talk. John Unit, Signature Discovery. I was struck by how you could use your platform to look for, polypharmacology within the GPCR family, because obviously there is some inbuilt redundancy in certain systems, and can you exploit that through structural modeling that you have recently just presented now?
Yes, so I think this is polypharmacology, basically promiscuity across multiple receptor targets, is just the other side of the coin of selectivity, which Ken asked about. So yes, we can predict this based on the uniqueness of binding site residues. We can do an overall comparison of the overall cavity in the central membrane domain. If you have a specific ligand and we can have a binding mode for that ligand, we can make a more specific prediction based on the binding site residues. With a manual effort coupled on that, we can of course point to selectivity hotspots that maybe you should interact with or introduce a steric clash with off targets.
Okay, thanks.
Thank you.
So final question, please.
Hi, Paul Insel. Hi, David. I wanted to ask a sort of a general question that I think has concerned a lot of people over the years, and that has to do with the G-protein coupling. And that is to look at the data that are used to actually generate the conclusion. And in many cases, one is using transfected cells that are expressed at much higher levels than the receptors are expressed at much higher levels than endogenous cells. So I'm curious, has that been taken into account in the analysis that you did regarding G-protein coupling?
Yeah, very important question. At least I want to mention that in the extent they could get hold of physiological agonists, they use those as the tool compounds, which is of course one parameter of resembling the native system. But then yes, overexpressed systems, yes. We did a normalization of data, and I think that's as much as we could do. We couldn't change the fact that data was using overexpressed systems. I'd love to see validation, maybe on a subset of receptors and couplings in more native tissues, and see how that compares. If we can make a systematic correlation, maybe we can also extrapolate that to the entire data set.
Yeah, one suspects that you'll get different answers when you do
Yeah,
that.
yes, and also tissue dependence. So with different tissues having different concentrations of the transducers, you see different selectivity profile across tissues.
Okay, thank you very much. I'd like to close the session. Please join me to give a big applause again to David Gloriam.